All right, in the interest of time and content, I want to thank everyone for joining. My name is Matthew Rose. I am a member of the New York COVID City Working Group, and I am proud to welcome you to this webinar today, put on by the COVID City Working Group, our amazing colleagues, friends, and the wonderful folks at IPAC, and our friends from Treatment Action Group. If you have been following this series, you know that the series from Columbia has brought you some of the most up information we've had on what's happening in science and on the medical side for COVID-19. And we think that this is a very needed topic to have discussion on vaccine. And today we actually have a great time to have the topic, since we are discussing a literal vaccine option, and one that's data might tell us more about some other vaccine candidates who should be releasing data by the end of next month. So figuring out the landscape is going to be very important. And we have two experts today to help navigate you through that, as well as tell you kind of where we're at and what is the state of play. First is Dr. Kathleen Neuzil, who directs the University of Maryland School of Medicine's Center for Vaccine Development and Global Health. Throughout her career, she has conducted clinical and epidemiological studies on vaccine prevention for preventable diseases. And at the global nonprofit path, Dr. Neuzil was instrumental in the introduction of rotavirus, HPV, and Japanese encephalitis vaccines. Second, we will hear from Dr. Holly Jennis, who's a business biostatistician working on design and analysis of vaccine studies with a particular expertise in HIV prevention and vaccine science. She also develops and applies statistical methods for evaluating biomarkers and risk predictions and optimizing treatment decisions. So we have both great content on the development side of what happens, how do you get these things through with a person who has a whole lot of history, and then is how do you evaluate, which is also going to be equally important. With that, I will pass it over to Dr. Neuzil.
So thank you, Matthew, and let me just take a minute here to share my screen. I'm very pleased to be here today. As people might imagine, there is no being ahead of the game in today's COVID vaccine world. There's no preparing talks and slides in advance. I think we've been, COVID has certainly been on the front page of the news every day for the last eight months or so, and fortunately now the vaccine news is dominating, and I see this as a good thing. So I'm going to outline a bit about the complexity of vaccine development, and then go through the current landscape of vaccines, and then Holly is going to get into some more specifics. We're just going to start here with where we were as of yesterday globally. You know, generally for many vaccines, we have to justify them based on burden of illness. And I think we can all agree that there is no justification needed for a COVID-19 vaccine. We desperately need a vaccine. We're at 50 million plus cases, 1.2 million plus deaths. This is the leading cause of death by any single pathogen in the world this year, and we're not close to the end of the year. What we're not used to seeing on these global WHO maps is the Americas, and specifically the US, leading the way. So we know that in addition to morbidity and mortality, we have been economically devastated, and communities in this country have been devastated. Our children are suffering from not going to school. And again, we have many non-pharmaceutical measures, but we certainly welcome vaccines and therapeutics. Now, I won't show a lot of virology, but I do think it's important to understand this one slide. And this is the SARS-CoV-2 spike protein, which is critical for viral entry. So on the left here, you actually see an electron micrograph of the virus, and you see where this virus gets its name. Corona equals crown or circle of light, and you can see there that this does look like a crown. And in fact, all of these projections here are a glycoprotein, the spike or S glycoprotein, which is shown in a schematic here. And here you see it binding to the host cell membrane. And that binding of the spike protein is incredibly important to initiate infection. And so the idea with developing a vaccine is that if we can make antibody to this spike protein, we can block the fusion of the viral membrane with the host cell membrane, and we can prevent or ameliorate infection. And in fact, all of the current vaccines in development in the United States and most of the vaccines in development throughout the world target this spike protein. So let's look at the usual vaccine development process, which is a lengthy, risky, and expensive process. You know, generally we identify the target. As I said, we build a case for burden of disease that it makes sense to invest in a particular vaccine. Then we look at preclinical development. We try to find the right antigen. We do animal studies. And then we move in a very deliberate way from phase 1 testing in just a few dozen people to phase 2 to very large clinical trials. In terms of optimizing manufacturing, we generally don't even start doing that until at least phase 2. That's a huge investment to manufacture an optimized product. And again, doesn't make a lot of sense until you are relatively certain that a vaccine is going to work. So how have we been able to reduce this usual time of 15 to 20 years for vaccine development down to 12 to 18 months? And I think in many ways, the naming of Operation Warp Speed has worked against us. And I hope that Holly and I assure you that we are not compromising on science. We are not compromising on safety. That in fact, it is the science, the resources, and the principle of assuming success at every step that have allowed us to greatly accelerate this. So on the science, we knew a lot about that S protein from SARS-1 and from MERS that allowed us to very quickly, within days, from getting the sequence of that S protein, have it in a stabilized pre-fusion form to begin vaccine manufacture. Without that basic science, without that 2 decades of work on S proteins, that would not have been possible. Similarly, our manufacturing has been improving over the last decade. In terms of resources, ordinarily you would never, as I said, begin to optimize manufacturing while you're still in phase 1. But that's exactly what we have done. And most of the resources have come from you and me, have been taxpayer resources in the United States. In other countries, it's been donor resources, the Gates Foundation, you've heard about CEPI, you know about Gavi. But this infusion of resources has allowed us to do things in parallel and not in sequence, and to assume success. So at the beginning of a phase 1, we are already planning for the phase 2, and we are already planning for the phase 3 to keep everything moving. So this is from the New York Times, who's able to keep up with things faster than I can, and perhaps this vaccine tracker has changed even this morning as you're watching my talk. But this looks at all the vaccines that are currently in the different phases of development. You see at the end there, we have 0 approved, but certain countries, not the United States, have approved vaccines for early or limited use. China, for example, Russia have limited use vaccine approvals. So let's just look, this is a slide that I made adapted from this very nice New England Journal article by Nikki Laurie at just some of the different platforms. So there are these DNA and RNA platforms, and the Pfizer platform, which is making the news, is an RNA platform. And in terms of speed of manufacturing, you can make these vaccines very quickly. And that's why they were the first that were into human testing, and likely in the US, the first where we will have an efficacy signal. And that is because what we do with these vaccines is we take the viral sequence and we essentially wrap it in a lipid coat. And I'll explain this a little bit later. But all you need to get started on manufacture is the viral sequence. Similarly, these viral vector vaccines use a virus to deliver a viral sequence. So that can be done quickly. The tried and true vaccines for which we have licensed products, influenza vaccines, hepatitis B vaccines are protein-based. Live attenuated vaccines, measles, mumps, rubella are all live attenuated vaccines. Those take longer. We have to find a substrate. We have to be able to make the protein. We have to be sure it is pure. For live attenuated vaccines, we have to make sure they're weak, but not too weak. And I think we would agree the idea of putting a live attenuated SARS-CoV-2 vaccine into humans would be a little scary unless we were sure again that it was properly attenuated and wouldn't revert to wild type. So these live attenuated vaccines work extremely well, and measles is a great example, but take a very long time. Now, these vaccines that take longer are tried and true vaccines. They have licensed platforms and they can be scaled. So if we need 7 billion people to be vaccinated with maybe 2 doses of vaccine, ultimately we want a vaccine that's scalable. What we are seeing at this point in time is there is perhaps one single dose vaccine, the Janssen vaccine. We should likely plan that these will be 2-dose vaccines. So Holly is going to talk a bit about this, but in the US, we've really taken a platform type approach where there are commonalities across these trials. And again, since Holly is going to discuss that in more detail, I'll just mention one that I think is particularly important. And that is for the Operation Warp Speed vaccines, which is all the vaccines except for Pfizer, actually. There is an NIH-convened Data and Safety Monitoring Board. So this is an independent board, and it looks at multiple manufacturers' vaccines. So if there are rare side effects or signals, this is a real advantage, both in terms of independence, but also in terms, again, of learning about these vaccines and assessing them. So in the US, and in fact, in most of the world, these are the leading candidates with some exceptions. So the Moderna and the Pfizer are mRNA vaccines. We have adenovirus vector vaccines. And then coming a little bit further behind are the recombinant protein and adjuvant vaccines. So as I explained before with these mRNA vaccines, we take this RNA, we take the sequence of that spike protein, we embed it in this lipid coat. It can then get injected into our muscles as part of the vaccine. Our muscles take this up, they read that genetic sequence, they make the viral S protein, and then our body sees that as a foreign antigen and mounts an immune response to it. Of course, this isn't infectious. We're not giving the whole virus, we're just giving that spike sequence. And while we've gotten used to science by press release, and that's what we saw yesterday, I can assure you that the early phase 1 trials for both the Moderna and the Pfizer mRNA vaccines have been peer reviewed and have been published in the highest quality journals. So these are the initial safety and immunogenicity results. The efficacy results are not yet peer reviewed. So this is just one slide summarizing the mRNA vaccines in the interest of time. But there is some good news here. So we see binding antibody responses after a single dose, but it takes 2 doses to give neutralizing antibody response. So what I mean by that is actually functional antibody that stops the growth of the virus. And if you look here, this is actually the Moderna vaccine. This was presented publicly at ACIP and is available in those articles I showed you. Again, we don't see much neutralizing antibody after the first dose, but quite robust responses after the second dose of vaccine that fortunately is preserved across age groups. We don't always see this. We sometimes see immunosenescence with vaccines, but we see the 3 different age groups and good antibody response. Now, we don't have a correlate of protection. We've only known about this virus for less than a year. So the best we can do is compare the antibodies that we're seeing from vaccines with a range of convalescent sera from people who have recovered from COVID-19. And as you can see, what we see with this particular example with the Moderna vaccine is the neutralizing antibody we are seeing after 2 doses of vaccine is as good or better than the antibody we're seeing in people who are recovering from COVID. We also know that in non-human primates, these vaccines are highly protective, and we've also seen monoclonal antibody results that are positive. So all of this is reassuring that targeting the spike protein is going to be effective. So we were actually the first to vaccinate as part of the Pfizer mRNA trial. That was only May 4th, 2020, and here we are 6 months later with early phase 3 efficacy results. Now, again, I don't know much more than the rest of you know from reading these articles, from looking at the publicly available protocol, and from looking at the Pfizer website. I can say that this level of efficacy is typically not seen for respiratory virus vaccines. I've worked on influenza vaccines for 25 years, and we would be dancing in the streets with this level of efficacy for an influenza virus vaccine. So what does this mean, and what do we know? We know that Pfizer intends to enroll 43,000 or 44,000 participants, and they're almost there, and nearly 39,000 have received a second dose. We know they're giving this vaccine in 2 doses, 21 days apart, and their primary outcome is efficacy against confirmed COVID-19. So you have to be symptomatic, and it has to be PCR confirmed. So this is laboratory confirmed symptomatic disease. So a fairly classic endpoint for respiratory virus trials. We've seen in the press release, they had 94 cases occurring 7 days after the second dose. Follow-up is ongoing. And again, they have made their protocol public, so I encourage any of you to go to their website and look at the details in the protocol. How do I evaluate this? This is certainly excellent efficacy. Yes, it may be against mild disease, but generally it's harder to get high efficacy against mild disease than serious disease. Yes, it's short-term efficacy, and of course it could decline over time, but at least we're starting high. We're not starting moderate and expecting a decline over time. And really, this is good news for the world, because this is proof of principle for a vaccine that targets spike protein, which almost all of the vaccines in development target spike protein. So what has the FDA told us? Well, the FDA has told us you need a minimum efficacy of 50%. So far, Pfizer is well above that. That in order to get these endpoints quickly, we're looking at large sample sizes, and Holly will go through that a little bit more, that we should look at the CDC definition of disease, which Pfizer and the other manufacturers had, that we should be sure to enroll those at high risk for disease, older age groups, chronic conditions, minorities. And again, if you'd like to go to the Pfizer website, you can see the percentage of all of these. And we also know that emergency use authorization is a possible pathway. And Pfizer cannot go for emergency use authorization quite yet because they haven't met this bullet here. The median follow-up duration of at least 2 months after completion of full vaccination regimen. So again, it will likely be several weeks, as they said in their press release, before they could even file for an EUA. At that time, we expect the case counts to be even higher because unfortunately, the epidemic is still raging in the United States and the other countries where these trials are ongoing. So I'm not going to say a lot about these other vaccines, just a couple quick slides. You know, the viral vector vaccines use a similar mechanism to the mRNA, but instead of delivering the sequence in a lipid coat, you're delivering the sequence in another virus. You're using a virus vector to deliver that S protein. And the protein-based vaccines are simply injecting the spike protein with or without an adjuvant, usually with an adjuvant, into our muscle cells. So this is just to, again, reassure you that phase 1 and 2 results have been peer reviewed, are available in the scientific literature, and the approach is the same. For the adenovirus vector vaccines, we see neutralizing antibody. For the AstraZeneca vaccine, the Oxford ChIP-Ad, after 2 doses, comparable to convalescent sera. The Janssen vaccine is the only one where they're proceeding in their phase 3 trials with a single dose based on seeing neutralizing antibody after a single dose. And similarly, if we look at the protein vaccines, the Novavax vaccine, really great neutralizing antibody, takes 2 doses to get there, takes an adjuvant to get there, but again, really remarkable early results with this vaccine. Now, the vaccines have also made the news because of adverse events. And again, you have all seen this. What does a clinical hold mean? A clinical hold is an order issued by the FDA to the sponsor to delay a proposed clinical investigation until an independent review can be done. So the AstraZeneca safety events were reviewed by a DSMB and the FDA, and the trial is now again proceeding. So we like to see that holds and pauses in trials are a sign that actually science is working and we are doing very close safety follow-up. Now, safety isn't easy with this vaccine. You've heard a lot of talk about vaccine-enhanced illness and what if that neutralizing antibody wanes? And so we will have to follow these people very carefully. And you know, with the vaccinations occurring in a short time frame with relatively short follow-up, you know, we're going to see efficacy before we have much safety follow-up. But the idea here is that the clinical trials give us robust follow-up, but we know that safety surveillance continues. It continues after the vaccine is licensed. There will have to be phase 4 studies, pharmacovigilance studies. And so again, the strong science is the starting point, but at least here in the United States and many other countries, we never stop assessing vaccine safety. You know, we're not yet into special populations. We know that children and pregnant women can be harmed by this disease. So once we have safety and we're starting to see the safety and now early efficacy signals in adults, you will see these manufacturers moving into children. Pfizer already has moved down to age 12. And again, work must be underway quickly for how we are going to distribute vaccines. A vaccine on a shelf never prevented a single disease. This is not going to be easy. Everybody knows the cold chain requirements of these mRNA vaccines requiring, you know, shipment on dry ice, -70 to -80 degrees. These will not be easy problems to overcome. And, you know, we're also starting to think about the national academies and the ACIP, the WHO SAGE, which I'm part of, who gets vaccine first. I refer you to these public available documents from SAGE, prioritizing use of vaccines in the context of limited supply. We will absolutely be in a supply constrained situation 4 months, if not longer. And so again, these value frameworks for allocation and prioritization of vaccine are important. Unfortunately, the United States is not part of this commitment and call to action for COVID-19 Tools Act Accelerator and for the equitable access of vaccines. Again, I'm personally a member of the WHO SAGE, but we're really looking forward to hopefully upcoming decisions for the United States to rejoin the WHO efforts and get on board with the Act Accelerator and COVAX. You know, this isn't going to be easy. If you look at lower middle income countries, this is a study we did here at Maryland. They just don't give adult vaccines in much of the world. The focus is on children and to some extent pregnant women. But this will be new, trying to deliver 2 doses of vaccine to older adults and to adults with chronic conditions. And I also refer you to this analysis by Ortiz and all, showing again that globally we must be prepared from the perspective of cold chain capacity, and also just the perspective of human resources and how many people we have to deliver vaccines. This is monthly vaccine doses per vaccinator. And look at the difference in Africa because of the shortage of healthcare workers compared to the Americas. So in summary, I believe I went a few minutes over. We absolutely need safe and effective vaccines, not a single vaccine, if we are going to have enough vaccine for the 7.5 billion people in the world and likely needing 2 doses. We should be reassured by the robust pipeline of promising candidates. We should be reassured by the safety systems and the processes that are in place. And as we saw yesterday, and this slide was written before that, these studies are large. They may reach these efficacy targets early. So we absolutely need to plan to properly resource our distribution and uptake plans. So thank you very much. And I will turn it over to Holly.
Thank you very much, Kathy, and Holly, welcome. If you'd please share your slides.
All right. Okay. Oops. All right. So thank you, Kathy. And I'm going to go through some more specific considerations, specifically focused on the trials, the efficacy trials that are in planning or underway under the Operation Warp Speed umbrella in the U.S. These are the trials that I'm most familiar with, but to varying degrees, many of the statistical considerations that I'll try to outline, apply broadly to COVID vaccine efficacy trials. But I'll highlight the approaches that have been taken for the Operation Warp Speed trials. So as Kathy mentioned, there are 5 trials under the Operation Warp Speed umbrella that are either in planning or underway. And this timeline is, was current as of about last week, and it is rapidly changing day by day due to, issues like, when does enrollment actually start, uncertainties around how long enrollment will take, and uncertainties around how long it will take to accrue the requisite number of primary endpoint cases over the course of the trial. So these are kind of approximations given the information that's at hand at the time that the figure was made. But you can see here, again, the 5 vaccine platforms that are being evaluated under the Operation Warp Speed umbrella. The Pfizer vaccine is another efficacy trial underway in the US, but is not formally part of Operation Warp Speed. So what's illustrated graphically on this slide is the rough timelines for these trials, all already enrolling or slated to begin enrolling before the end of this year, with readouts in terms of efficacy shown using the end of the purple line there, sometime in the beginning of 2021. So what I'll try to go through is at a high level the statistical design attributes of all these trials, and highlighting the attributes that are shared across these trials. As Kathy mentioned, there's been considerable effort towards developing harmonized trial designs to facilitate at the end, once we have data on multiple vaccine platforms, cross-protocol analyses to learn about differences in safety and efficacy amongst these vaccines, and to combine the data across the trials to enable analyses to be done against endpoints and events that occur with relatively low frequency in any one trial. So in general, these trials are enrolling somewhere in the neighborhood of 30,000 participants. The populations that are being sought for enrollment are adults 18 and over with no upper age limit, who are determined to be at appreciable risk of both SARS-CoV-2 acquisition as well as subsequent COVID-19 disease. And importantly, the trials are not screening based on laboratory testing for evidence of prior infection and enrollment, for a variety of reasons, including the imperative to generate safety and efficacy data for a broad population of adults, given the necessity to roll out in an efficient fashion vaccines to protect the global population. However, the trials are enriched as much as is possible for individuals who are high risk of acquisition and disease based on knowledge of predisposing risk factors for those conditions, including older age, comorbidities that are associated with worse outcomes of COVID-19, as well as race ethnicity, where for the US in particular, given the disproportionate impact of COVID-19 in underrepresented minority populations, there's a commitment to enroll underrepresented minorities at or above US demographic frequencies in these trials. And the participants who are enrolled are randomized in either a 2-to-1 ratio or a 1-to-1 ratio to receive vaccine or placebo. Sometimes this randomization for some of these trials is stratified based on risk criteria such as age or pre-existing conditions. And the choice of a 2-to-1 or a 1-to-1 randomization of vaccine or placebo has basically the merit of a 2-to-1 randomization ratio is it's slightly more statistically efficient, but the primary rationale for using a 2-to-1 ratio is to improve power for discovering immunological correlates of protection, which is something that Kathy mentioned. I'll come back to at the end of my slides to discuss that objective. Generally speaking, the trials are planned to follow participants for up to 2 years post last vaccination to assess long-term safety of these vaccines. And the common primary endpoint across these trials, as Kathy mentioned, is virologically confirmed symptomatic disease. So this is a schema that shows the general approach to collection of primary endpoint data, as well as other key secondary endpoints, and the schedule of visits and collection of specimens across these trials, where what's shown here in the schema is a hypothetical 2-dose regimen. As Kathy mentioned, 4 of the 5 vaccines under the Operation Warp Speed umbrella are 2-dose vaccines. So the participants are enrolled and followed for safety up to 2 years post last vaccination. The primary endpoint is virologically confirmed symptomatic disease and is ascertained using weekly contacts with participants to query them about COVID-related signs and symptoms, where if a participant reports those conditions, a pre-specified list of conditions specified in the protocol, then they're brought in for laboratory testing using PCR. And if confirmed to be infected based on PCR, coincident with having this protocol specified list of signs and symptoms, they constitute a primary endpoint case, a virologically confirmed symptomatic disease case. And also shown in this schematic is that at baseline and at a scheduled set of visits post-enrollment, there's blood collected from the participants, and this enables us to assess the immune responses that are generated from the vaccine. It enables us to assess retrospectively which of the participants had evidence of prior infection with SARS-CoV-2 at enrollment, and it also enables us to assess seroconversion, SARS-CoV-2 seroconversion over the course of follow-up, which may or may not be associated with symptomatic disease. Individuals who are diagnosed with COVID over the course of follow-up are then enrolled into a post-COVID diagnosis follow-up schedule shown in the graphic on this slide. And basically that entails, daily assessment of signs and symptoms and severity thereof throughout the duration of presentation of symptoms, and every other day, and then decreasing frequency of swabs that are collected for ascertainment of the extent and duration of shedding of virus, which is a key secondary endpoint relevant for understanding transmissibility of infection. So the graphic showed on this slide illustrates summarizes and illustrates the structural relationships amongst the primary and key secondary endpoints that are collected across all of these trials. And basically we can conceive of these endpoints. So some of the individuals over the course of follow-up will be diagnosed with SARS-CoV-2 infection. Some of those individuals, perhaps about 60% of those individuals will have symptoms associated with infection, whereas perhaps about 40%, based on current CDC estimates, may exhibit asymptomatic infection. And then among those with symptomatic disease, symptomatic infection, COVID-19, a small fraction of those individuals will exhibit severe disease. Perhaps 5%, a small fraction of those with symptomatic disease will be severe, and then the vast majority of those with symptomatic disease will be non-severe COVID disease endpoints. And so all these trials will collect all of these endpoints to enable a comprehensive assessment of the full spectrum of vaccine effects. But the primary endpoint, the common primary endpoint across all these trials is shown in blue here. Virologically confirmed symptomatic COVID-19 disease. This is, as Kathy mentioned, a classic endpoint for other trials of vaccines against respiratory infections, an endpoint that has clear relevance to the individual as well as to population public health. Infection endpoint shown at the top of this slide is also a highly relevant and significant endpoint, has relevance to understanding transmissibility of the infection in the population, and would perhaps be the holy grail, a vaccine that completely blocked infection. But given experience with other pathogens, and in particular respiratory infections, where there has traditionally been higher efficacy against symptomatic disease as opposed to against complete blockage of infection, the primary endpoint of symptomatic disease was chosen as primary endpoint. Down at the bottom of this slide, the severe disease endpoint, this is an endpoint that perhaps has the most relevance to an individual, but which, as I mentioned, is expected to occur at a very low frequency in the trials. So again, all these trials will collect the full spectrum of endpoints to enable a full and comprehensive assessment of vaccine efficacy against all these endpoints, but the primary endpoint is against virologically confirmed symptomatic disease. And I'll note that related to the FIDSA results released yesterday, that 90% vaccine efficacy, 90% or greater vaccine efficacy is against this symptomatic infection endpoint in the middle of this slide. So shown in this slide is a graphic illustrating the time course of these studies. Again, for a 2-dose regimen. So these trials are all what is called event-driven trials. This is a classic approach taken in clinical trials where there's uncertainty about the incidence of the primary endpoint. So what that means is that the primary analysis of vaccine efficacy will occur at a time point that is not defined in calendar time, but rather is defined based on information. And in particular, information in terms of the total number of primary endpoints that have accrued in the trial. So what's been done is to determine the total number of primary endpoints, those COVID-19 disease endpoints that are needed to evaluate vaccine efficacy. And that's about 150 primary endpoints if there's a 2-to-1 vaccine to placebo ratio, and 170 endpoints under a 1-to-1 vaccine ratio of vaccine to placebo. And so the primary analysis will occur when those 150 endpoints have accrued. And the timing of that is somewhat uncertain because it's impacted by what's going on in terms of the epidemic in the broader population, as well as impacted by the level of vaccine efficacy. But the trials have all been sized at this large sample size to ensure that under conservative assumptions around the incidence of COVID-19, that primary analysis, when 150 events have accrued, is likely to occur within at least 7 months of trial start. And unfortunately, given the exploding epidemic in the US in particular and in other regions around the world, it's likely that for many of these trials, that primary analysis will occur much sooner than 7 months of trial start. But once that primary analysis has occurred, there have been plans laid to continue follow-up of participants, ideally blinded follow-up, through to the end of month 24 to assess long-term safety of these vaccines and to evaluate vaccine efficacy against severe COVID-19 and other endpoints that are likely to occur at a lower frequency in the trial. So a few details about how efficacy will be evaluated, at least in terms of the primary analysis of these trials. So the classical measure of vaccine efficacy in a vaccine trial is defined as 1 minus the ratio of the rates of infection of the primary endpoint in the vaccine versus placebo groups. So a 90% vaccine efficacy corresponds to a hazard ratio of 0.1. And this is classically assessed using a proportional hazards model that allows for a separate baseline hazard of the endpoint in different study sites and in different randomization strata, reflecting considerable heterogeneity in terms of the incidence of COVID geographically, as well as across subpopulations. For all these trials, the primary analysis of vaccine efficacy will be conducted among participants who are retrospectively determined to have been baseline negative for SARS-CoV-2 at enrollment. This is the subpopulation of individuals in whom it's believed there is the greatest potential benefit from vaccination. And this primary analysis will be conducted in what's called a full analysis set. This is all participants who are enrolled and who have received at least one dose of the vaccine. Most of these trials will do their primary analysis where only COVID-19 events that occur 15 or more days after the last dose of the vaccine are counted towards that primary analysis. And this is to give the vaccine the greatest possibility to have effect by allowing the immune responses induced by the vaccine to mount before infection events or before disease events are counted. However, I'll point out that importantly, all these trials will include as key secondary analyses the fuller set of vaccine efficacy assessments based on all participants who have been enrolled, counting all events post-enrollment, and without regard to the timing of when the event is diagnosed. The success criteria that have been form the basis for these study designs entail a point estimate for vaccine efficacy against COVID-19 disease of at least 50%, at a lower bound on the 95% confidence interval around vaccine efficacy that rules out 30% or lower vaccine efficacy. So this is a success criterion that has been adapted or adopted following guidance from the FDA, as Kathy mentioned, and as well guidance and consensus coming out of the WHO. This is the success criteria for the minimally acceptable success criteria for put forth by the WHO, corresponds to their target profile, product profile for COVID-19 vaccine. So I'll mention that there's considerable work underway to anticipate for and capture potential or predicted population impacts of COVID-19 vaccines, and this is work by colleagues that's looking at the population impact of a COVID-19, a hypothetical COVID-19 vaccine that meets this minimal success criterion, achieves a 50% reduction in COVID-19 disease over the course of a 1-year vaccination period. And this is potential projected population impact of such a vaccine when rolled out in the population of King County, Washington. And you can see on the left here the cumulative the cumulative deaths in King County, Washington, that are projected to occur absent a vaccine program over the course of 2021 in the black line, and the cumulative number of infections in the black line on the right-hand side, absent a COVID-19 vaccine. And then what's shown here is the projected impact of rolling out such a vaccine to 45% of the population over the course of 2021. That has, that achieves a 50% reduction in COVID-19 disease, and that has a varying level of efficacy against preventing infection and against reducing onward transmission of SARS-CoV-2 infection. And what we see here is that there's even with a vaccine that is minimally effective, reduces disease by 50%, there is a large impact of rolling out such a vaccine in the population, resulting in a 40 to 69% reduction in the death rate over 1 year and a 33 to 70% reduction in the infection rate over 1 year. Again, for a vaccine that has a relatively modest impact on COVID-19 disease, relative to what we're hearing coming out of the Pfizer trial yesterday. So back to the design of these trials, how do we identify the sample size? How did we come up with 30,000 participants, 30 to 60,000 participants to enroll across these trials? Well, it's basically geared off of what this success criterion is. We determined that 150 primary endpoint cases are needed to have 90% power to detect a 60% vaccine efficacy and to establish success criteria for a vaccine that has 60% efficacy. That's the number of endpoints that's required if a 2-to-1 vaccine to placebo ratio is used. And then we work backwards to figure out the sample size that is needed in order to have a high probability to accrue 150 primary endpoint events. So, for example, if we anticipate going in that 90% of the individuals enrolled who will be baseline SARS-CoV-2 negative, and assuming very conservatively a 1% 6-month incidence of COVID in the placebo arm, this yields a required sample size of 30,000 participants. Importantly, all these trials wrap into their design and implementation pre-specified plans for monitoring for early evidence of potential harm or enhancement of COVID-19, non-efficacy or weak vaccine efficacy of the vaccines, as well as efficacy of the vaccines. And in particular, there's continuous monitoring of potential harm of the vaccines to very aggressively generate a signal if there's any evidence of a potentially increased rate of disease or severe disease in the vaccine versus placebo arm. There's generally speaking 2 interim analyses to detect early evidence of absent or weak vaccine efficacy to ensure that result could be delivered to the field in a timely manner and preserve resources for other trials and other vaccines. And as well, there are, generally speaking, 2 interim analyses to evaluate and detect early evidence of efficacy of the vaccine to permit rapid decisions, regulatory decisions, and rollout of that vaccine. Generally speaking, these interim analyses are using conservative O'Brien-Fleming boundaries. So as reflected on this slide, all these interim analyses are based on not calendar time, but information time. Their occurrence is dictated by how many events have accrued over the course of the trial, and therefore the timing of these interim analyses is uncertain, in advance of the trial, and is not known except to a small group of individuals who are examining the interim data. So as Kathy mentioned, there is a common data safety monitoring board that is overseeing all the trials under the Operation Warp Speed umbrella, that is charged with looking at the interim trial data on a regular basis, evaluating it in its entirety in terms of protecting participant safety and trial integrity, and in particular relating the evidence that is accruing to these pre-specified boundaries, defining efficacy, non-efficacy, and potential harm. And so that data safety monitoring board, that independent group of experts, can then is facilitated, sending signals if needed around whether or not any of these boundaries have been crossed, based on the data from the trial itself, and also based potentially on the data that are emerging from any of the other trials in the Operation Warp Speed portfolio.
Holly, if you can please up so we can have some questions.
Yep. Okay. All right. So I think I'll skip this. I think what I'll do is just highlight another key component of this program, which is development of immune correlates. So this is a major focus of the Operation Warp Speed program, and it's really critical, we believe, for accelerating the development and licensure of COVID-19 vaccines. One area of recent interest where the importance of this is illustrated is if we can generate evidence around the immunological immune response generated by these vaccines that predicts the level of efficacy associated with these vaccines, really the holy grail of vaccine research for any pathogen. This would rapidly accelerate vaccine development. It would enable future trials of candidate COVID vaccines to be done using several hundred persons instead of tens of thousands of persons. Particularly important if and when an early vaccine is shown efficacious, which would tend to necessitate non-inferiority trial designs, which would be extraordinarily large if a clinical endpoint study is mandated, as opposed to much more feasible, much more rapid if an immunological biomarker was assessed. And as well, understanding correlates of protection is really critical for bridging vaccine efficacy to new populations and not included in the initial efficacy trials, for example, adolescents and pregnant women. So there's a intense interest in this, and there's key structures of the Operation Warp Speed program that have been set up to facilitate this immunological immune correlates program, which include harmonized trial designs, common endpoints for assessing the immunological responses to vaccine, and a common statistical group and data sharing agreements to facilitate these analyses. I think really unprecedented the level of work and the pre-planning that's gone into doing this and ensuring. So I won't get into the specifics here in the interest of time, but just back to the timeline, in order to ensure that these data that can be generated, just a couple of months after the completion of each of these trials, so that we hope there will be firm evidence around the immunological correlates of protection sometime in the middle of 2021, again, rapidly accelerating continued development of COVID-19 vaccines. So I'll stop there to leave time for questions. Thanks.
Thank you. Thank you very much, Kathy. And thank you, Holly, as well. I'm Wafaa El-Sadr. I'm Global Director for ICAP at Columbia University, and I'll be moderating the discussion. So please keep sending your questions. I have a question here. Let's start with you, Kathy. And the question is, as you mentioned, all of the vaccines are targeting the S protein. And so this is good news because we have the signal from the Pfizer vaccine. So what are the implications? I mean, do you think this bodes well for all the other vaccines as well? And on the other hand, somebody raised the question as to whether that makes us particularly vulnerable if, for example, there's a mutation that develops that could then mitigate the effects of all of the vaccines. Kathy?
Yeah, so really good question. I mean, it makes sense, and with many respiratory viruses, we want antibody to that protein that's instrumental in binding and attachment. So similar with influenza. And while we've seen some strain differences with coronavirus vaccines, I can definitely assure everyone this is not influenza. We don't see that kind of change in the spike protein. So we have looked at, with these trials, neutralizing antibody to some of the other circulating strains, and it appears to be preserved. There's a new strain circulating in Europe that has people somewhat concerned, and the testing has not been done yet for that strain. So I think it's something, again, coronaviruses are not influenza, but it is something that we will need to continue to monitor. And in the same way we've done this with influenza vaccines, there are some vaccines in early stages of development that target multiple proteins or conserved areas. So this is a real learning agenda at this point with a new virus.
Thank you. Thank you. And maybe another, Kathy, question for you before I move on to Holly is concern about that children are not involved in most of these studies, the vast majority of the studies. And is that of concern and particularly because of the issue of transmission within households and families and so on?
Yeah, it's a great question. Again, we know with other respiratory viruses that children usually shed more virus for longer periods of time and have behavior patterns that facilitate transmission of virus. And so many of us, me included, are really advocating for earlier rather than later testing of children. We in the United States have almost as many deaths in children due to COVID as we have had deaths due to influenza in children for the last several years. And so the idea was just what's the right time to move into children. We want to be sure we have the safety data in adults. And as I said, with the good immunogenicity and the good efficacy signal, I think you will see these trials age de-escalating and moving into children over the next few months.
Thank you. And Holly, a question for you is, if the primary endpoint, as you mentioned, is symptomatic disease, COVID-19 disease, what are the implications at a population level of having a vaccine that prevents disease but does not prevent infection in terms of transmission in a community?
Considerable population impact of a vaccine that prevents disease, but not infection. Although I would say that importantly, all these trials are also collecting endpoints, infection endpoints that are not necessarily symptomatic. Any individual who seroconverts over the course of the study would be captured by each of these trials, and so we will be able to assess the efficacy of these vaccines against preventing infection, which, as you allude to, is critical for understanding the full spectrum of impacts of a vaccine at a population level in terms of reducing infections and onward transmission of virus. I think in terms of the effect of the vaccine on reducing transmissibility of infection to other individuals, potentially mediated through reduction in viral replication, reduction in viral shedding. We will not be able to assess that with these trials, given that there's only viral shedding data collected post symptoms among individuals who are symptomatic. So that will be a later phase of research. But again, we will be able to assess vaccine efficacy against infection, which has major implications for population transmission. But as I tried to articulate using the modeling work, even a vaccine that has a relatively modest effect on preventing disease is projected to have considerable population impact in terms of reducing the burden of transmission.
And I think some of the concerns are arising from, like we're now noting that in some parts of the world, the asymptomatic, the proportion of individuals with asymptomatic diseases appears to be much higher than even in Europe and in the US. A question for you, Kathy, is what are the implications of Pfizer, if the results hold and Pfizer goes for an EUA, what are the implications for all the other ongoing studies? Can you speculate on this?
Yeah, that's a very challenging question and something that we've probably been talking about for months within each of these individual trials. And it's dependent on certain factors. It's dependent on the degree of efficacy. Right now, we're seeing a very high degree of efficacy, but we don't know in what populations that efficacy is occurring. So we need to see more of the Pfizer data. Is this being driven by the young, healthy population and we still won't have an answer in older adults, or will we have an answer across the age spectrum and across comorbidities? It will also be dependent on supply of vaccine. We will be in a supply constrained situation early on. And so again, the ability to perhaps continue some of the trials in groups in whom the efficacy hasn't been shown, or in groups for whom there is no vaccine available because the vaccine is only, for example, going to be recommended for healthcare workers or older adults, may be possible. But we should be actively preparing also to vaccinate to the placebo group in these trials. And Holly explained some of that, and they can be followed over time. And also having to do comparative trials. And, we've done this in the influenza field, high dose versus standard dose. It takes large numbers of people, and it takes longer, but these trials can be done.
Great. And a question that's been raised as well by several individuals is the whole ethical issues of how do you scale up once you have a vaccine in hand, who should get it first, whether it be within the United States or globally. And you mentioned one approach by National Academy of Medicine that's been proposed. But how do we ensure that we are doing what's ethically and morally right in terms of deployment of an effective vaccine?
Yeah, these are difficult questions. And again, in the United States, we are fortunate in some ways that we have so many leading candidates and that the government is buying vaccines. So this will not be, I go down to CVS and buy a vaccine because I have greater privilege and access. This will be more controlled by the government and likely prioritized in our usual mechanism, which is the Advisory Committee on Immunization Practices and the CDC. It'll be a bigger challenge in the rest of the world. And you saw some documents where SAGE is beginning to have these discussions. They're looking at perhaps a distribution of vaccines initially to all countries. That's about 3% of the population. So this is really very little vaccine going to each country and then providing some guidance by the countries. Another way we could look at this is vaccine could go to where the epidemic is the worst and where the epidemic is raging. The problem with that is that could disadvantage those countries with poor surveillance. So none of these priority schemes are perfect. The key will be to get as much supply as quickly as we can.
And maybe one last question. I know we're running out of time. We have plenty of questions and little time, but for you, Holly, is a question about, as you know, the controversial approach of the challenge, the design of challenge vaccines. So can you comment briefly on your thoughts on that kind of design for vaccine research?
I think what I'll comment on is simply the what can be learned from a challenge trial. There's critical
Explain it very briefly what it is.
Okay. So a human challenge trial is a setting or a study in which healthy individuals are enrolled, given randomized to vaccine or placebo, and then experimentally challenged or exposed to the pathogen, so that in a very controlled setting, and treated therapeutically if they become infected. So it's a type of study design where, because of it, by its controlled nature, there's a lot of scientific insights that can be generated by such a study, because the investigators know that the timing of exposure to the virus and can study very intensively the early course of infection, the immune responses that are generated by the vaccine, the type of events that are prevented by the vaccine. but it's challenging in terms of navigating the challenging ethical issues and clinical safety issues in that context. So I'll let Kathy tackle those very difficult issues. But you know, there are important scientific insights that could usefully be generated from a challenge study. One, Wafaa, you alluded to, is understanding the impact of a vaccine on infectiousness of an individual who does become infected despite having received vaccine. That's a sort of insight that could be generated very readily from a challenge trial. And so, you know, the fact that it appears that some of these trials are beginning to become feasible and acceptable in certain settings and in certain populations could accelerate the development of COVID vaccines.
Thank you. And Kathy, give you the last word on this issue, if you have any other comments on the challenge.
So in terms of the challenge studies, yeah, I mean, we've done respiratory virus challenge studies here. I personally feel more comfortable doing them when a rescue therapy is available. They really could help with this duration of immunity question. I think their greatest limitation is that you pick the youngest, healthiest volunteers for a challenge study. So if they behave differently than the populations we're seeing at highest risk for morbidity and mortality, then what we learn is going to be limited.
Thank you. Well, thank you both very much, our speakers, Doctors Kathleen Neuzil and Holly Janes. Thank you for joining us today, and thanks to all of you who attended this webinar, and thank you for your many questions. And I'm sure this is not the last webinar that we're going to have on COVID-19 vaccines, and as well as many, many other interesting topics. So have a good day and stay safe and stay connected. Thank you.
Thank you, all of you, very much.